Hello, everyone. I welcome you here today and thank you for coming for this webinar session brought to you by Foodline UCC on opportunities and challenges of cold membrane filtration in dairy processing. So here is today's agenda. Today we are going through the brief introduction of membrane filtration, its performance in terms of flux and fouling phenomena, and then I will try to define warm and cold filtration and compare some of the linked advantages of flux advantages and disadvantages associated with cold and warm filtration. Going further, we will go through cold filtration research trends. And finally, we will take a closer look at opportunities and challenges associated with cold filtration. And hopefully at the end of the session, I will have enough time to respond as many as questions as possible. So without further delay, let us go through membrane filtration briefly. As you can see here, the projected global market for membrane filtration and microfiltration is growing as there is a rising awareness of health conscious consumers and a demand for nutritious enriched foods are growing, and so is the demand for dairy protein market. So here we see the chart consisting of particle sizes for various milk components and the range of membrane technologies that are applied in dairy industry. Two of the most common technologies that are used are membrane filtration and ultrafiltration. MF is largely used for fractionation purpose and UF is used for concentration. So here are some of the common applications of MF technology. We can largely classify them in three categories, which are bacterial reduction. So for example, for extending the shelf life of milk or cheese milk. And second is milk protein fractionation, used for making fractions of milk into micellar casein and serum proteins. And third, last is the fat removal for making protein isolates. Here you see the ultrafiltration and its common applications, which are largely protein concentration for making either MPC or WPC. And the second is protein standardization, most commonly used for making for cheese milk. So here is the typical process applied in dairy industry. The process begins from whole milk and can move towards various individual processes. Here are some of the highlighted processes that are relevant for membrane filtration. So for example, MF can be applied for manufacturing either MCC or to use casein fraction as a base for cheese milk. UF is applied to manufacture either MPC or WPC, etc. So two major formats of membrane applied in dairy industry are either organic, which are polymeric membranes like polyether sulfone, and they are largely used in the form of spiral wounds. And the second is the inorganic membranes that are ceramics, and they are mostly tubular in tubular formats. So the ceramic membrane is most cost intensive than polymeric membranes, but they are highly stable at harsh temperature and pH conditions, which are used for membrane cleaning process. In dairy industry, the cross-flow membrane filtration configuration is preferred. The accumulation of solids in dead-end filtration, as you can see here, often results in lower flux, which is often not ideal for dairy processes. So here you see the flux membrane filtration flux, and the membrane filtration is largely judged by its performance in terms of flux. In any filtration process, flux will decline with time, and there are largely three stages of flux decline. So the first stage is the rapid drop, where the rapid absorption of protein occurs on the membrane surface, and it occurs within a few minutes of filtration. Second stage, where the flux drops continuously and the further deposition of protein takes place. In the third stage, the flux drop becomes slow and steady, and the additional protein deposition on the surface takes place, and the cake layer that is formed becomes overlap. So the stages of flux decline are clearly the results of fouling. Therefore, we can look slightly more into details of individual fouling mechanisms that can take place one after another or even simultaneously. So the first is adsorption, which occurs when protein adheres on the membrane surface or within the pores. It can narrow the channels of the membrane or form cake layer on the surface. These are reversibly bound proteins and can be removed with cleaning. Second is cake layer formation, which occurs where particle aggregation take place, and they form bridges and piles. And it consists of large and small particles, so wide range of particles, and it can get compressed over time, but it is reversibly brown bound, so it can be removed by cleaning. And so the third one is pore blocking. So it happens when the pore is plugged by particles slightly larger in size than the pores. And it decreases the number of pores available for the fluid flow. However, these are also reversibly bound and can be cleaned by cleaning. And finally, then is the in-depth fouling that happens when excessive transmembrane pressure is applied, which forces the particles deep into the pores. It is largely irreversibly bound and is difficult to remove with membrane cleaning. So here we see the range of temperatures applied for membrane filtration. Warm filtration can be defined as an operation carried out at temperatures ranging between 45 and 60 degrees centigrade, while cold filtration is carried out mostly between 4 degree and 20 degrees centigrade. So there are associated advantages with warm filtration. For example, higher permeate flux, lower fluid viscosity, sharper protein fractionation. However, there are also disadvantages or challenges associated with it. For example, higher microbial growth, several severe membrane fouling, or very rapid flux decline. In cold filtration, the advantages are lower microbial growth, less severe membrane fouling, and it also offers an opportunity to develop innovative ingredients, for example, beta-casein concentrate and isolates. However, cold filtration also has its disadvantages too. For example, lower permeate flux, high fluid viscosity, and protein loss in permeate. So, for example, loss of casein when we do cold MF into bay permeate. So all the pointers I highlighted here are identified from the research articles I reviewed to conduct the survey on cold filtration research trends. For that, I surveyed 8 scientific journals that are largely relevant for publishing dairy science research. I searched a keyword, microfiltration, and surveyed the article most closely linked with skim milk microfiltration. So I found in total 109 articles, out of which highest number were published in Journal of Dairy Science, followed by International Dairy Journal. I compared them further on the basis of cold and warm filtration, and this is how the trend looks like. Warm filtration research was largely published in dairy food journals like Journal of Dairy Science or International Dairy Journal, while cold filtration was more prominent in core membrane and technology journals like Membrane and Separation and Purification Technology. Ceramic membrane was largely used in all studies, with the exception of Membrane Journal. Most of the MF studies were either carried out for cheese manufacture, MF performance studies, to study the fractionation with different pore sizes of membrane, different types of membranes, etc. And thirdly, for microbial removal. So we can see here that the cheese manufacture studies were dominant in Journal of Dairy Science and International Dairy Journal, suggesting the trend towards application of membrane filtration technology for cheese making, largely in United States and the Europe. So the popularity and trends towards cold filtration were also validated by many stakeholders, including equipment vendors, membrane manufacturers, dairy processors, and end users such as ingredient formulators. However, despite the popularity, it is important to highlight that there are associated unknowns to cold filtration, such as the filtration performance, ingredient quality, fouling, protein permeation. It has given researchers like me an opportunity to answer to these unknowns and share that knowledge with wider community. So from here on, will look into each of these four questions that are linked with cold filtration and highlight the findings and its linked implications. So in this slide, you see the flux performance at different processing temperatures. So for filtration, I used ultrafiltration membrane of 10 kilodalton to make 3.5x concentration retentates and study the flux performance during the filtration run. As you can see here at 5 degrees centigrade, flux was lowest, but it declined slowly. It took about 120 minutes to reach 3.5x volume concentration when operated at 5 degrees centigrade, which is almost double the duration that took for 50 degrees centigrade. So clearly in terms of flux, warm temperature offers better performance, but only initially, which then declined very rapidly. So here you see the chart for viscosity of fresh feed. Clearly, the feed viscosity was highest at 5 degrees centigrade, despite the equivalent content of solids as other feeds. At cold temperature, casein micelles have lower hydrophobic interactions, and colloidal calcium phosphate begins to solubilize within serum phase, which causes the water to uptake within the micelle, and that causes the swelling behavior of the casein micelles. And this swelling leads to increase in fluid viscosity. So as we see the trend for the feed, similarly, UF retentates with about 12% protein had highest viscosity at 5 degrees centigrade, while comparable trends were observed for 50 degrees centigrade and 20 degrees centigrade. This high viscosity is an unavoidable challenge to achieve high protein concentration beyond certain point. For example, in this case, only 12% protein or about 3.5x volume concentration could be achieved with UF on its own without any additional diafiltration steps. However, despite these challenges of lower flux and high viscosity associated with cold filtration, it still shows potential to manufacture high-quality protein ingredients. Plasmin is one very stable proteolytic enzyme in milk, which can survive pasteurization treatment, and it is highly detrimental to ingredient quality because it can do proteolysis of proteins in longer run of time. So here we see the effect of processing temperature on plasmin activity in UF retentates and feed. As you can see, the highest plasmin activity was measured at 50 degrees centigrade and lowest at 5 degrees centigrade, both in feed and retentate. However, concentrated retentates had lower plasmin activity than feed. So why is that? This could be due to the molecular size of various enzyme components of plasmin system, which consist of inhibitors as well as activators apart from plasmin itself. Since the UF membrane is very tight, which is what I used is 10 kilodalton, so which is way smaller. So plasmin inhibitors may be retained by UF membrane due to their molecular size. Also, whey proteins are regarded as plasmin inhibitors, which was also concentrated with UF. So that could reduce the plasmin activity even further. But overall, we can see here that the lowest activity was observed in 5 degrees centigrade UV-free 10 days. So one important method to measure extent of fouling is by measuring normalized water permeability of membrane before and after membrane cleaning. So immediately after you have run with milk to achieve 3.5x volume concentration, I rinse the membrane with water and measure the NWP. And then I followed the cleaning steps with alkali and warm water. And after cleaning, I measured the NWP once again. So this bar chart represents the NWP before and after cleaning of the membrane. I would like to draw your attention to the bar on the left, which indicates NWP before chemical cleaning. So as we can see here, at 5 degrees centigrade, the NWP remained highest, even though filtration time was higher to achieve 3.5x volume concentration, which means milk was in contact with membrane surface for longer time than when operated at 50 degrees centigrade or 20 degrees centigrade. This indicates that there were more pores and channels available for water passage after cold filtration was carried out. It also suggests that the deposit layer built up on the membrane after cold filtration was less compact. So this suggests less intense membrane fouling at cold temperature. So here we see the level of protein permeation in permeate after milk MF at range of cold temperatures from 4 to 20 degrees centigrade. As we know, beta-casein solubilize from colloidal casein phase into serum phase on cooling, which then permeates through MF membrane during filtration. So here we can see this temperature-dependent beta-casein permeation was highest at 4 degrees centigrade and lowest at 20 degrees centigrade. This beta-casein enriched whey permeate introduces an opportunity to develop innovative dairy ingredients with great market potential. So as I mentioned before, there are great potentials of cold MF applications like beta-casein enriched whey ingredients or isolation of beta-casein to make concentrates or isolates, or to reduce beta-casein to make cheese milk with less bitterness. However, despite these applications, major application of MF is to target milk protein fractionation, where casein can be completely retained in retentate and whey permeate, fraction remaining highly pure. However, this is a technology challenge for cold MF, and where we cannot stop the casein release into permeate by simply modifying the operating conditions like TMP or filtration viscosity. But it requires the intervention on casein level to restrict the release of casein monomers into serum phase while cooling milk. So therefore, one possible solution is to apply enzymatic cross-linking to casein micelles with enzymes like transglutaminase, which is commonly applied in dairy industry for manufacture of yogurts, cheese, and even milk protein powders. So transglutaminase acts by targeting lysine and glutamine amino acids in the protein chains and induces cross-linking by making isopeptide covalent bonds, which are strong. Cross-linking action turns monomeric proteins into higher molecular size oligomers and polymers, which also changes the conformation and stability of the protein. So in this case, of the casein micelles. Since casein contains both amino acids, lysine and glutamine, and it has more open structure than whey proteins, it is regarded as an excellent substrate for this enzyme action. So in order to study the effect of transglutaminase cross-linking on casein partitioning during milk fractionation, approach we used was to change casein micelle environment by altering the surrounding temperature and simultaneously inducing the transglutaminase action. Here you see two environments casein micelles were exposed to. Warm temperature of 40 degrees centigrade for 90 minutes and cool temperature 5 degrees centigrade for 24 hours. Simultaneously to this, I added the enzyme to initiate the cross-linking action. So if we focus our attention to feed warming, we see cross-linking taking place in micelles and making stronger covalent bonds within the micelles. However, there are some locations where enzyme might not react, which leaves potential protein fractions to behave as they are natural, meaning they can dissociate later if feed is once again cooled. On the other hand, while feed is cooling, some protein is out in serum phase, moving freely. So in addition to the cross-linking action on casein micelles that we saw in previous case, here it also takes place with freely available casein monomers. So enzyme will either cross-link the monomers back with micelles, changing their mobility permanently, meaning they will not be able to dissociate again when feed is cooled later. And second possibility is that the enzyme will cross-link monomers while they are in serum phase. This will also change their mobility and structure so they cannot return to the micelles and remain available in the serum. So here we see the protein profile of control feed and cross-link feed. It is evident here that cross-linking has taken place in both TA and TB feeds, which means uh, cooling feed and warming feed. Casein bands are not very visible in TA, indicating extensive cross-linking here. However, we see some beta casein available in TB bands, which indicates that not all the casein was cross-linked with transglutaminase. Cross-linked proteins can be seen located above at higher molecular sizes. This strongly suggests cross-linking and formation of large-size oligomers and polymers. So, as it was confirmed that the feed was successfully cross-linked, we conducted cold MF of this feed at 5 degrees centigrade. The filtration cycle was run in recirculation mode for 90 minutes, meaning permeate was returned back to the feed batch. The permeate samples were collected at various intervals and analyzed later. So here this bar highlights the total nitrogen content proportion out of total solids measured in permeate samples. As we can see, comparable proportions of nitrogen content in TA and TB permeate of only about 13 to 15%, which is very low than control with about 70% of total nitrogen out of all solids. So this suggests very low protein content in TA and TB permeate as compared to control. So to further support the results, we can look at this image and understand how permeate samples look visually when they were warmed from 5 degrees centigrade to 55. All three permeate look similar at the start at 5 degrees centigrade. But when we begin to warm the permeate, beta casein begins to demonstrate its self-association behavior as indicated by rising turbidity of the samples. So control has highest turbidity at 55 degrees centigrade, which indicates that it contains beta casein that can self-associate on heating to form aggregates. And in TA sample, the turbidity is very low at 55 degrees centigrade, which suggests unavailability of beta casein, which can self-associate to form aggregates. And similar to TA, TB also has lower turbidity than control sample, but it is higher than the turbidity of TA, which suggests that there is some beta casein available that has ability to self-associate into aggregates. So overall, this study suggests that there is a possibility that the protein release was limited into permeate by enzyme cross-linking. So this study demonstrates the potential of transglutaminase cross-linking to limit the casein loss in permeate during cold MF. With this, we come to the end of this session, and maybe a quick recap would help us remember what we covered today. So we covered a brief introduction of membrane filtration and key factors to judge its performance with flux and fouling. Further on, we compared the cold and warm filtration and highlighted the major opportunities and drawbacks of each technology. Then we looked into cold filtration, research trends, and stakeholders validated interest in this technology. Later, we reviewed various opportunities of cold filtration related to membrane fouling and ingredient quality, and its challenges associated with casein loss and possible technological solutions to tackle those challenges. Overall, we see there are associated advantages and disadvantages to cold and warm filtration. We need to strike a balance to achieve best potential of these technologies. So for those who are interested to read further on today's topic, here are some open access publications from our group, Food Ingredients Research Group, and some external publications as well. With that, I take this opportunity to thank everyone for their patience and time in listening to this session today. And now I invite our audience who might have the questions that I would try my best to answer as many of them as I can. So thank you once again.